my notes on that one. No.
You're one of the people that are like, I know, we take this for a minute, because,
Yeah, because it'll be online.
sorry, I was checking my emails. Apparently my computer has sent our IT department a help message. Something's wrong with its hard drive. Okay, no name is on it. So I have to tell them to come take a look at it. That's funny. I never thought they could send a message. I need help. I know, self-aware. I should be more careful in my office. All right. Okay. So I'm not recording it because, again, I have to pause it and all of that if you guys want to record it. So for review, chapter 8 again is the big one. Start by making sure you understand double-stranded DNA, the structure of it, the terms associated with it, anti-parallel, complementary. How do you make the sugar phosphate backbone? How are the two strands connected? So you have two sugar phosphates on the side, and then you have, where is it? So you have two sugar phosphates on the side, and then the hydrogen bonds in the middle. Be aware of that. So a strand direction 5' to 3'. So where the phosphate is attached is to carbon number 5. And then the 3' carbon is where the next nucleotide is going to come to, and covalent bonds are made. Rules about DNA. DNA is read 3 to 5. DNA, whenever you're making DNA, you creating a nucleic acid based on a template on the DNA, you are making complementary new strands, new RNA, new DNA. It's complementary. And since it's complementary, it's going to be anti-parallel. So you read 3 to 5. You always make it a 5 to 3' direction. Why? Because the enzymes have to attach to that 3'. If you know those, then logic follows what DNA kind of does. For DNA replication, you can understand what is chromosomes, what is the structure of chromosomes. When this animation that I have is like 5 minutes or so, it's pretty good. It's a video. She really goes over homolog chromosomes, the 23 pairs in humans, how it shows the chromosomes, the location of the genes. So if really that bothers you still, take a look at it. She even goes over the concepts that I think haploid, diploid, and things like that. All right, so then you start thinking about replication. So genotype, phenotype, know what that is. Replication, like I said, just go and, sorry, these are preliminary stuff. So DNA replication. Just know the order of your enzymes. DNA helicase, I have gyrase, topoisomerases, then you have DNA polymerases, and finally after DNA polymerase you have ligase that I don't have there. But the most important ones are the 5 ones that I said, the order of it. Helicase, primase, DNA polymerase 3, DNA polymerase 1, DNA ligase. All right, come up with a way for yourself to remember it. Replication always starts at where? What do we call it? That place? Origin of replication. So when the cell is ready to do origin of replication becomes active. What attaches to origin of replication and starts? Helicase. And it does what? The term we use? Unzips. So separates the hydrogen bonds. You will have stabilizing proteins in the animations. I'm not going to ask for that much detail, but DNA will, if you let it go, it will collapse on itself. It will try to repair the hydrogen bonds. So in replication, we will put proteins to keep them apart. Then the next enzyme is primase. Why do we use primase? First of all, what kind of enzyme is primase? RNA polymerase. So we are making DNA. Why am I using an RNA polymerase?
Because DNA polymerase only needs a 3' carbon to attach to it. So
Any DNA polymerase, whether it's 3 or 1 or 2, they need a 3' carbon. So again, the example that I use, if Inga and I are the complementary nucleotides, DNA nucleotides, it is the job of DNA polymerase to say, okay, Sina, you're complementary to Inga, so I'm going to make hydrogen bonds between you. But it doesn't do that. Why? Because it wants another nucleotide right here before me with a free 3' carbon so it can make a covalent bond between me and the previous nucleotide. And in DNA replication, you don't have that. So it's like DNA polymerase refuses to start and you don't have a nucleotide because if DNA polymerase doesn't put down a nucleotide, what are you going to do? But RNA polymerases don't have that issue. RNA polymerase come and see Inga and they go, all right, Sina prime, RNA form of Sina is a complementary for that one. I don't care to make this covalent bond. I'll just make the hydrogen bonds and I'll make covalent bonds from here on, read and put other RNA nucleotides. So we use primase again to have a 3' carbon. RNA primer is about 10 to 15 base pairs. Primase falls off, then it's 3. DNA polymerase 3 comes, attaches to the 3', reads the template, puts complementary nucleotides, and goes on. Eventually, you can't have RNA in DNA. So those primers need to be removed. That's the job of DNA polymerase 3. You don't have the issue of 3'. Why? Because, so let's say this is my old strand, and here, so A, T, C, C, G, G, T, T, A. I'm just creating a weird, whatever, strand for myself. 3 to 5. So here is where I have a all right. So my primer, let's say it's here. So it goes A, U, U, G, A, U. All right, that's the primer that was here, and it's going to be 5 to 3, and then it's going to be DNA from that point on. All right, and this is the previous primer that was here, and then the DNA strand. So A, C, C, G, G, A, T. Remember, DNA polymerases won't create that bond. So this is your lagging strand and it had a primer here. So if it wants to replace this primer, what it actually does, DNA polymerases can actually go to an existing DNA and say, okay, this is the primer. I'm going to go a few base pairs above in the DNA, make a cut in the sugar phosphate backbone there. So now, remember this is 5 to 3, right? So attached to the 3' of that C nucleotide DNA polymerase attaches there and starts moving forward. As it moves forward, it cuts that one, chews it up. So removes it and places a new C there, a new A there. So it's going to remove these, and as it goes, it does the same replacing. C, A, does continue the same thing, goes here, makes this to A, makes this to T, T, G, A, T. DNA nucleotide, it will go, even into the DNA that was there, then fall off and say, that's good enough. The rest should be okay. So that's how DNA polymerase goes and replaces the primers. And the last thing is DNA ligase. Because they leave that part, so they don't make that sugar phosphate covalent bond, so the 3' of the last nucleotide they place, they don't make a covalent bond with the phosphate of the next nucleotide that is there. It's, again, issues that they have. DNA ligase comes and says covalent bond, fixes that. All right, now you know the order. What's the difference between leading and lagging? When you have DNA replication in DNA replication fork, one template, the direction of reading follows the helicase, so therefore the direction of replication follows the helicase. So you put down one primer, and then DNA polymerase 3 comes and reads continuously as helicase moves. In the other one, again, because of the anti-parallel structure of DNA, it's going to be called a lagging strand. In the lagging strand, the DNA replication and reading of DNA template is done the opposite direction of the helicase, so fragmented, multiple primers, multiple DNAs, Okazaki fragments, and then after DNA polymerase 1 removes the primers, they're no longer Okazaki fragments. They're going to become continuous. That's it. Know the terms semi-conservative. Semi-conservative means the DNA, newly formed DNAs will have an old strand and a new complementary strand. So like I said, this one is very good. Go over that one if you want. How do they know something is an old DNA strand? By methyl groups. So they know that the old strand has methyl groups. Once everybody's happy with regards to the new DNA, they will add the methyl groups. DNA polymerase 2 that we don't talk about much, that one is the major editor. Its job is like an inspector, constantly checks the new DNA. Repairs, they can repair the same way that DNA polymerase 1 removed primers. All right, then I said forget about replication, put that aside. When we go to gene expression, that's the function of DNA. Has nothing to do with DNA replication. Function of DNA is express its gene. When the gene is expressed, a protein is made. The process of protein synthesis has two portions, transcription and translation. So first, since DNA is very important, you don't let everything to come and read the DNA, your recipe, and like ribosomes, tRNAs, everything come there. So what you do is you make a transcript of it. That transcript is mRNA. Every gene, we said, has a promoter section starting it and a terminator stopping it. So to make that, when the gene is ready to transcribe, I just said you don't need to worry about the regulation. Magically, the promoter sequence becomes active. That allows for one enzyme to attach. And that enzyme doesn't even have any formal names or anything. We just call it RNA polymerase. Why do we use RNA polymerase? Because the ribosomes can use it in us eukaryotes. It comes out of the nucleus, so DNA is protected. We want to make sure DNA is DNA, so any copy is RNA. RNA polymerase attaches to an active promoter, unzips DNA as it moves along. It will choose a template that goes 3 to 5 from promoter to terminator, read and make a complementary mRNA, and once it gets to terminator sequence, basically there is a sequence in terminator that causes the RNA polymerase to kind of lag. It creates a loop in it and causes it to fall off. So it knows it's the end. It falls off. RNA polymerase is gone, mRNA is there. For prokaryote, this happens in the cytoplasm. For eukaryotes, it happens in the nucleus. For prokaryotes, it's mature, it's ready to be translated. For us, it needs to be processed. So we need to remove the introns, which are junk pieces of DNA. Those are removed. Exons are attached to one another. Then we add a 5' cap and a 3' tail. Now our mRNA is mature. Comes out of the nucleus and goes and it gets translated. So understand codons. Codons are made up of 3 nucleotides. Each codon means one specific amino acid. Our genetic codon is universal and redundant, meaning because with the 3 nucleotides, there is 64 possibilities. Cell and nature has taken care of that, and some amino acids may have multiple different codons stating the same amino acids. In the genetic codon, it is on the mRNA. That's where codons are, and that's the only place codons will be, on the mRNA. Then you also have 3 codons that tell you the stop, and they are not associated with any amino acids. And you will have 1 codon that says start, but is associated with amino acid methionine also. So technically, for 20 different amino acids that we have, or the bacteria have, we have 61 different codons. Total is 64 codons, but 3 of them are stop, and they do not code for any amino acids. They just say stop. Remember? All right. Then we have the process of translation. Again, ribosomes are involved in that. tRNAs have the anticodon and associated with specific amino acids. So the small subunit comes, attaches, makes sure that mRNA is nice and straight. The first or initiating tRNA that is against the first AUG code, which is the start codon, comes and interacts, and then the large subunit sits, puts the first tRNA in the P site, the second tRNA comes now at the A site. Once they are there, the large subunit allows for the amino acids to form a peptide bond. The amino acid at the P site goes on top of the amino acid at the A site. tRNA becomes discharged, ribosome moves 1 codon down, so then it goes into the E site. From E site is going to come off that empty tRNA, go find its amino acid. Continues until it reaches stop codon. No tRNA associated with that. There is actually a protein that would sit there, but if you just say no tRNA associated with it, I'm fine with it. And that's when everything dissociates and the protein goes to be done whatever way that it needs to go. So for them, this process is much faster because they can actually transcribe and translate simultaneously. For us, multiple different things have to happen. By the way, not that I want you guys to know, but somebody emailed me and asked me, and I was like, okay, I'll let everyone know. They were like, so does that mean that we only have methionine at the beginning? No. The simplest answer to this complex thing is there is 2 tRNAs for AUG. There is one that is a little bit of a different tRNA, and that's the one that is initial tRNA. It knows this is when it's AUG, this specific tRNA comes with methionine, or F-methionine for the bacteria. But the other AUGs have a tRNA that it's just attached to methionine. So even for bacteria, the rest are methionine. So they're a little bit different. So it just happens the tRNA, the cell knows this is the initial AUG, it uses that one, and this is the additional methionine. So you would have the methionine in there. But again, it's a little bit more complex problem. It's cellular biology, biochemistry, right? You guys don't need to worry about that. In fact, many of the first methionine for many proteins is clipped off. They remove it when the protein is folding. So. All right, differences, make sure you understand those. Mutations is basically when you have a permanent mistake in replication process. Then we talked about point mutations here. So base substitution, insertion, or deletion. Point means one nucleotide is affected, that nucleotide can be substituted, or you can have a nucleotide that is added extra, or a nucleotide that is not put there, so deleted. Then we said the substitution can create neutral mutations. That's when the same event-based substitution results in a codon that says the same amino acid, so nothing changed really for us. Or it could affect one base substitution, could affect only one amino acid and create, in this case, missense versus nonsense, all right? Missense is when that amino acid is changed. Now, depending on where it is and that polypeptide, it could totally change the protein. It could be really bad, or it's functional but not that great. But again, it's just one amino acid. Nonsense is when one amino acid is suddenly changed to stop. So no amino acid. But deletion and addition, we said those are worse because they cause frame shift. From that point of mutation, if it was deleted or inserted, every codon downstream from that point is wrong. Every multiple amino acids will be affected. Beneficial is just if it helps with survival of the fittest, natural selection. And then we talked about combination gene transfers. Don't worry about crossing over. But in microbes, especially bacteria, understand that we can have horizontal gene transfer, which in eukaryotes we don't see. Transformation is when they pick up DNA from the environment, from a dead bacterial cell. Somehow, a fragment of DNA from the environment ends up inside their cytoplasm, they soak it up. Transduction, a virus, bacteriophage, actually takes and transfers a bacterial DNA, and conjugation was the one that they create conjugation with. And that's all I wanted you guys to know. Yeah, so biotechnology, don't worry about it. 9 and 10 are really simple. So for 9, again, understand these terms. What are they? So sterilization is removal of all microbes. Either you stop their reproduction or physically remove them. That's sterilization, except for prions. So know these terms. Then microbial death for us is when we stop the reproduction. They can affect all these different parts. So have an idea about this chart, what is most susceptible and what is most resistant. High level, intermediate level, low level, again, just a basic understanding of it. Have an idea about effect of heat, moist heat versus dry heat. So this is Seidel. Cold temperature, refrigeration, freezing is static. Lyophilization or desiccation. So dehydration, these are again all static. Filtration could be sterilization. So if you have proper filters, you can remove microbes. You can use it for air, for liquid. Osmotic pressure is a pretty good one. Again, can have, in some cases, like somewhere to static to sidal. So it's pretty good. And then radiation. Ionizing versus non-ionizing. They affect DNA. Ionizing is obviously better, and it's the newer techniques that we use for food prep. And then chemicals, like I said, just know the difference. Have an idea about like phenols. Lysol is an example. It will probably mostly affect proteins, plasma membranes. It's intermediate to low level. Alcohol could actually be intermediate. It could be very good, but then it evaporates really fast. So that's not so good. It's good and it's bad. So halogens, these are chlorides, betadine, iodine, bromine. So they will affect by kind of like oxidizing and affecting the proteins. Then you have oxidizing agents, hydrogen peroxide. Again, it's good for cleaning an area, but for antiseptics, not necessarily always it's good. When you have catalase and you have, you're releasing, you have damage in your blood, you have catalase. Surfactants are soaps, detergents. Detergents are much better, and they can actually damage lipids and proteins, heavy metals, copper, silver nitrate, aldehydes. They can be high level. They can sterilize. They interact with all the side groups, so functional groups, and gaseous agents. You know, those are the ones that we don't want to use as much because they are dangerous, but they can be poisonous. So have an idea about those two, and I think that's as much for this chapter. And then we go to chapter 10, which I went over today. So know the terms chemotherapeutic drugs, antimicrobial drugs, prophylaxis or prophylactic. Know what that means. Don't worry about the history. Then selective toxicity, understand what that term means. Therapeutic index, what is that? And then for cell wall, I said 4 methods. Either beta-lactam ring, like penicillin, that attaches to transpeptides, prevents crosslinks. Or there are drugs that go specifically affect the crosslinks if they have 2 alanine amino acids next to one another. Or they can have drugs that prevent NAG and NAM secretions completely, so they don't make even the chains, NAG and NAM. And finally, ones that affect mycolic acid, all right, for mycobacteria. Then not much in fungal area even, we don't have much, and the others don't have a cell wall. Protein synthesis, affecting 70S specifically, so translation process in bacteria. Either they will change the shape of 30S or the small subunits, so it causes misreading of the mRNA. Blocking the A site of the large subunit prevents the protein synthesis. Preventing the enzymatic function of the 50S, so peptide bonds are not formed. And the last one is anchoring the ribosomes to the mRNA. Again, no need to know the name of the drugs. Cytoplasmic membrane, not a whole lot. The only one I gave for antifungal, ergosterol, but even that is not a great one. Then metabolic pathways. This is where the sulfa one is important. So you have sulfa drugs are analogs of PABA. They prevent folic acid productions in bacteria. In us, no effect because we consume our folic acid. We don't make it. Nucleic acid synthesis, again, not a whole lot. DNA gyrase can be targeted. Polymerase, RNA polymerase in tuberculosis. ACT is a good one example of it, affecting reverse transcriptase or being analogs of some of the nucleotides. And antivirals, mostly they prevent them from getting in to the cells or preventing them from coming out. There is interferons, which we'll look at when we talk about immune system. Interferon is something we make and actually stops the process of protein synthesis in our healthy cells temporarily because a virus is there. So it's kind of like an alarm that the infected cell sends, and your healthy cells go, ooh, a virus might be here. So they stop protein synthesis, but we can't have that for a long term, otherwise our cells die. That's the pains that you get in your joints and all of that when you get the flu, is because your body produces interferon. Some antiviral medications have interferon in it. And that's why the patients complain. They're like, oh my God, I constantly have joint pains, muscle pains. And then think about spectrum of action. Broad spectrums are not good. Now we like narrow spectrum. Efficacy, so obviously something that is easily used. MIC versus MBC, M-I-C-K-E-Y-M-O-U-S-E. So Mickey Mouse, MIC and MBC. Just have an idea what they are. That's how they measure them. Again, you won't be asked to explain it, but be familiar with it. Disc diffusion, obviously be familiar with it, and you're going to become very familiar after Wednesday's lab. Selective toxicity, and then resistance. For resistance, just understand it's naturally there. How they pass it on to others, either they survive an increase in number, and then they can pass to one another by horizontal gene transfer. So increase it in other bacteria that it's not them. And how do they do that? They pump it out, they change the target. These are the results of it. And multi-resistance versus cross-resistant, and then also how do we limit the appearance of. All right? So hopefully that helps you guys. On Wednesday, we're going to start chapter 11. Again, that will help with 9 and 10 a little bit more. All right, guys. That thing is still ported. We are 1:35. All right. I'll see you guys on Wednesday.